Okay, hello everyone and happy Monday. I think that we're now on the week 14 of the semester, so really approaching the finish line. I keep seeing on my screen that the signal for me is kind of going in and out. So for the purposes of keeping the session running smoothly, I'm just going to turn off the video, but I wanted to say hi in person, or at least with a face. And I do realize that it's the week before Thanksgiving, so a lot of people have a lot going on. So I really appreciate everyone who has joined today. Before we get too much further, though, what I wanted to do, there's a couple of things that I just need to make an announcement. And the first one is really more of an apology. If you have messaged to me and you have not heard back, please know that there's no ill intent or there's nothing mean behind that. For the past week or so, I've just been totally snowed under with various things going on at the college. For that reason, I am a little bit behind with messages. So if you have not heard back from me, I will get back to you. If it was about an extension, if it was about something that was time sensitive, I will take that into account and make sure that you get that extra time starting from when I respond to you. So please, please, please be patient, just like students. We also get busy. I think that for instructors, it's often a couple of weeks before students get busy. We get busy with preparing the exams and preparing things. But I apologize for that, and I will get back to each and every message that I've received. So don't feel that I'm ignoring you. The other thing, some of you have become very good about it, and just as a reminder, you can always resend the message to me. And if I keep seeing it at the top of my message list, usually that makes sure that that gets answered as the first one. But like I said, there's been no agenda. You guys have not done anything wrong. If you have not heard back from me, that is something that's I own that, and I admit it, and I apologize for that. And I'll get back to each and every single one of you as soon as possible. So having said all that, I also want to share the same calendar view that we saw earlier on. So last week I shared with you that these two last chapters that we're looking really this week and last week are kind of bundled together in a way that I prepared the learning module for weeks 13 and 14. So there is actually only one quiz for both chapters. We covered about half of the learning material last week. And even though I had it on the study guide, I do think that during the session that we had, I did share it verbally that I was not expecting you to complete the quiz by November 18th. What I've moved it to is November 23rd. And the quiz is actually made of only 10 questions. And there is no lab, so really should be quite a doable amount of work. What's important for me is that you get it all done before Thanksgiving so you can focus on enjoying the Thanksgiving with your loved ones. So I have set the deadline for the quiz covering chapters 14 and 15 for tomorrow, end of the day, so November 23rd. If you have already gotten the quiz in, I appreciate it. If you haven't yet, we will today actually go through all of those questions that might come up on that. So those were the kind of two most urgent things that I wanted to share about. I do also want to just point out, kind of have a little bit of look towards the future, that next week, when you come back from the Thanksgiving break, it's going to be our final week of actual teaching as such. And on the week 15, all I have for you for scheduled is an exam 4. So that covers the last 5 chapters or so. And we will, as always, take the time to go what might come up in the exam using a study guide. So we'll do that on Monday, a week from now. So we have a class in a virtual format, and I will be recording that as well. But I will not be, as before, we've used the study guides. You're now familiar with those. I will not be uploading the completed study guide. There will be gaps. So it's a good session to either attend or watch recording of that. In terms of future things, the final week of the semester for students is week 16. And since we have been taking exams quite frequently on this course, it's been and that's how I designed this course, that we take rather exams often and smaller area to cover those. So it's been my plan that on the final week, really you should be quite early on aware of where your grade has landed. And based on that, I will be releasing extra credit work. And you don't have to complete the extra credit work. But once you've had a chance to look where your grade might have landed after completing all of the assignments, that week is there for you really to get in extra work if it's not where you want it to be. I know many of you are taking these courses as a prerequisite for other things that you're working towards. So I want to give every chance to bring up those grades. I also am quite flexible towards end of the semester with opening any assignments you might have missed if you messaged to me, or retaking assignments. But what I would ask that we hold the fire from that until we're back from Thanksgiving break, simply because I don't want to have an influx of messages on the message system, because I still have some to answer. So all we're doing today is going through the final material in terms of the lecture for this course. And then tomorrow I'm asking you to complete the homework quiz for chapters 14 and 15. And once you come back from the Thanksgiving, then we'll wrap up the semester. But it really is designed to be low stress. And having said all that, I do want to point out that I have not scheduled a class for this week's Wednesday. So there is no formal teaching. And same goes for the next week when you're back from the Thanksgiving break on Wednesday, there's no formal teaching. I will be around in case someone has a question. I might not be logged into this room, but you can send me a message and I will jump in if necessary. But I think that those are the biggest announcements that I had. And before we jump into the actual material, I just want to take a moment in case anyone has any questions that you want to ask about what I've just shared, what I have failed to share, if there's anything else that's weighing on your mind. Like I said, I do apologize for the fact that I'm still catching up on some of those messages. We'll take an attendance in just a few moments, but do we have any questions about anything relating to the practicalities of this course, practicalities of this week's assignment and last week's assignment? I think I've seen a message that there was one quiz question, or it might have been an exam question that didn't grade correctly automatically. And all of those things I will go back, and whenever I go back, it will apply to everyone if there's something that's found out. So just wanted to share that as well. Well, I'm not seeing questions coming through. As always, I'll try to stay at the end a little bit longer. I don't expect this to be a super long session, but I will stay around in case you have any one-on-one questions. So having said all that, I think it's a good time for us to get to the chapter material. And on this chapter, chapter 15, we kind of shift our attention from talking of the generic DNA. And a little bit of this will be revision. So we'll discuss about this journey. Oh, hold on a second. We'll discuss about this journey of us going from genes, going from DNA, and how we can make something useful out of them. And often the output, if you wish, is the proteins. And I know that we touched upon this topic earlier on last week when we talked about chapter 14. So this week will be really kind of a reminder of those concepts and taking those concepts a little bit further. So we will have a look a little bit further at how does that all apply in real life use of the knowledge that we have about genetics. So with that all said, I do have a little bit of a quiz question. This is actually the last question on your this week's quiz, just as a way for us to take an attendance. So if you can, please post to the chat, what do you think is the correct answer? And bear in mind that there might be multiple correct answers. What is a gene? Is gene a section of a DNA? Is gene made of base pairs? Does gene code for a product? And if you post to the chat, that works as your attendance, and I will then, based on that, just get it recorded. Anyone who's worried about the attendance, really the big thing is that you attend at least once a week, and largely I take that through these class attendances. So if you don't mind, I would really appreciate you posting an answer to this on the chat. And if you don't know, you can just answer that I don't know, but I'm going to find out. I just need some sort of an answer on the chat to give you an attendance for today. And what we end up seeing, and what I think I'm seeing on the chat, is that all of these three statements are correct. Gene is a section of a DNA that codes for a particular thing. DNA was made of base pairs, so the gene, the section of the DNA, will also contain the base pairs. The base pairs are really the information part, if you wish, of the information that we're storing at the DNA. And gene codes for something useful. It might be a product. This product can show itself as a trait that we see. Often the products are seen through a gene coding for a particular protein. And these proteins are then often, I describe them as a workhorse of the cell. So all of those would have been correct answers. Thank you for everyone who posted an answer to the chat. And what I wanted to do was kind of to revisit the really core concept that we talked about last week and kind of expand a little bit on that. So some of the elements of this, I assume, will be familiar, and some of it will be a little bit of an extension from what we have discussed. So let's have a look of the journey from DNA to all the way to the proteins. And what we're seeing here initially is a section of the DNA. So you remember it was a double-stranded, double helix structure. And typically it would be this kind of twisted around itself. So for the purposes of us being able to look through this, I have kind of straightened it out, flattened it out. But we see the two strands, and they have these base pairs that are connected, or these bases that are connected to each other. So they are always match each one of those. But what we can do, we can actually open the DNA, these two strands, and make the two strands separate. And to do this in a lab setting, we often use heat to assist in this process. So by heating the sample, adding certain enzymes, we are able to go from this double-stranded structure to having it open so that other things that we want to happen will take place. So now we have opened the DNA, and we can do the process of transcription. You remember that transcribing referred to making a copy of the original template. So on this example, we have the original template in a green color, and we're making a complementary copy to that. You will see it on the yellow or orange color on this screen. This process, transcription, was a process that takes place within the nucleus. So you remember DNA never leaves the nucleus of the cell. And once we have made a copy of the DNA, the copy that we've made is now going to be RNA. So RNA was able to leave the nucleus. But we see that RNA can be found within the nucleus, within the cytoplasm, attached to many ribosomes, many other parts of the cell. So RNA really has unlimited access. We do also find RNA, of course, within the nucleus. And when we kind of take this information that we know at this point and we start thinking about the applications of it, that relates really to the topic of the day. And one of the methods that we'll be discussing about is called PCR. PCR stands for polymerase chain reaction, which is a technique we use in DNA sciences to make many copies of a section of a DNA. So we can use PCR to multiply the amount of the genetic material. So that will become something that we'll discuss in just a little while. Well, now we have our RNA. It has left the nucleus of the cell. It's traveled within the cytoplasm and found ribosomes. So what we're seeing here is a single ribosome. A ribosome is made of multiple parts. And what you'll see that the ribosome kind of scans through the RNA strand. And what ribosome does, it looks at 3 bases at one time. And based on those 3 bases at one time, it uses that code to identify which amino acid should be picked up from the cytoplasm. So there's going to be amino acids floating around. And it is really this coding, reading of the information that we have recorded on this RNA strand that happens at this site. And as a result of that, like I said, we're picking up individual amino acids. And these individual amino acids picked up based on that information of 3 bases at the time, they're brought and added to this chain that slowly forms out of the amino acids. And this process, of course, as you remember, was known as translation. So rather than just simply making a copy, which was transcription, translation is giving a meaning to the information that we have. So the meaning in this case is kind of finding the amino acids that the information codes for. And when we have a string of amino acids, as we see here, well, this string of amino acids actually makes up a protein. And if you refer all the way back when we first started talking about proteins on this course, one thing that you'll remember is that proteins could be studied in many different levels. We could study them based on their chemical formula, chemical structure, but we could also study them in plated sheets or even in a 3-dimensional shape. And this amino acid chain really folds into a protein that then is the workhorse of the cell, workhorse of implementing whatever those instructions that we had in the DNA are. So I hope that that kind of helps you to remind what we have discussed so far, and also works as a kind of a starting point for any extra information that we'll add to the story during today's session. And now that we have discussed about that, I do want to take a moment just for anyone who might have any questions about that, might feel a little unclear. If there's anything, this is a really good time to let me know, and I will definitely do my very best to answer. Well, again, if you have questions, don't hesitate. I'll try to monitor the chat, but you can also unmute yourself. So we have just looked through this journey from the DNA to making proteins that then implement things. I think this is a good chance for us to also play a little bit of a reminder from our last week's chapter. So I have a couple of questions that really relate to what we have already seen, but that will come up in your weekly quiz. So for that reason, I wanted to take a little bit of time to talk about those. Some of you might remember that last week we talked about Dolly the sheep. And I have a question here that's kind of a tricky question if you're taking quizzes or exams whenever there's the word not. So make sure that you notice that that question asks that which of the following 5 statements is untrue, is not true. So is it that the Dolly, the sheep, is an example of cloning? Is it that Dolly the sheep was made from an adult somatic cell? Was Dolly the sheep created in Tucson, Arizona? Did it take 277 acts to create Dolly the sheep? Or is it that the technique used for creating Dolly can be also used to bring back extinct species? And I do realize that it's a week from when we last met, but we did discuss this, and I'm seeing answers coming through. The true correct answer is that, of course, Dolly the sheep was not created in Tucson, Arizona. So all other statements about Dolly were mentioned in our session. And it is actually this example of cloning what we will be focusing today to a large extent. I do have another question about Dolly. How many mothers did we say that Dolly had? So you can answer either with a letter or a number. How many mothers did we think that Dolly had? And again, I really appreciate anyone who's posting, taking part. I've been saying this all along the way, but I do want to assure you that I am not just saying it, it will happen. I do give credit also for active participation. So I'll at the end sit down and count all the times that you took part, and you'll get points for that. And that gets added to your final score for this course. And good job, anyone who said that there were 3 mothers, if you wish. So one thing that we saw, that we used a cell that came from one mother, but we removed all the material, the genetic material from that cell. So that could be viewed, if you wanted to, as the first mother of the Dolly as such. So rather than having a cell where we have the nucleus, we simply removed all that material, genetic material. So we had a blank cell. Then we took another cell, and from that cell, we moved the nucleus, only the genetic material, into this blank cell. But you'll remember that after that, we did give an electric shock, which triggered the process of cell division. And this cell that we had generated now started dividing as if it would have been fertilized cell. And that cell got implanted into another sheep. So there were really 3 mothers involved, if you wish. So cell from one, genetic material from the second, and third one was the gestational mother that carried Dolly to the full term. So that might be a reminder from what we've discussed last week. So really, thank you again for everyone who took part. Really what I want to focus today is these applications of everything that we have learned about genetics. So we do much more than grow garden pea plants these days. And the field of genetic engineering involves inserting cloned genes into an organism. So we take genes that we're interested, then we want to make a copy of those genes and we insert them into an organism that's our target. And really, I think that that brings us to the term cloning. So whenever we're talking about cloning, we're talking about making identical copies. So the original source of the material is going to be genetically identical to the offspring, to the resulting cells that we're making. And what we end up finding is that when we study the genetics of humans, apes, other animals, even plants, the genetic code is nearly identical from one organism to another. There are some differences in the amount of the genes that we have, in the amount of chromosomes that we have, but really the method of coding this genetic information is pretty much the same from one species to another. And because it is pretty much the same, we can use this knowledge and these techniques that we're going to be talking about really to transfer genes from one organism to another. And they don't even need to be same species. And this might sound a little bit of science fiction to some of you, but I assure you that we actually already for quite a while have been using these techniques. We've been fairly cautious and fairly selective when we use these techniques, but it is not something that would be brand new or something that is totally out there in terms of being an alien concept to us. It happens and it's being used quite widely. To what extent and on what species? That's where there's a little bit of variation, and that's where some of the ethical discussions also come to play. So, one of the important concepts that I want us to look at today, and I want us to spend a little bit of time, is a concept of doing recombinant DNA techniques. So recombinant DNA techniques are sometimes abbreviated also as rDNA techniques. You know, in science, we love abbreviating everything that we possibly can. So what happens in recombinant DNA techniques is that we are producing an output, the final organism that comes from this, as an organism that has DNA from two different sources, two different organisms. These can be organisms of the same species, or this can be also organisms of different species. So you might ask that, well, how does that happen? That sounds a little weird. And we're actually going to see it to quite a great detail how that happens today. So that's one of the things that I want us to start with and consider. So what you're seeing here at the very top right corner is a section of a DNA. So we're seeing the double-stranded structure, and we're seeing how bases match. So there was this pattern that we saw that adenine always matched with thymine, guanine with cytosine, and so on and so on. So these were the complementary base pairs. Well, what we can do, we can identify a gene, a section that we're especially interested out of this DNA strand. Let's say that we're interested about everything moving onwards from the third last base pair on the right, shown on this picture. So what we want to do, if we're interested on a gene that had this pattern of GCG, and then continuing from there onwards, we want to cut that gene off because it's doing something that's useful for us. It's something that we really want to use, something that's coded into our genetic makeup and that's producing the output, the product that we're interested. Well, we want to cut that piece of gene off. And on this example, I'm only focusing on one end. We would do the same thing at the other end of this gene, but for the sake of keeping it simple, I'm only going to be looking at one end of this particular gene. So you'll notice that what we can do, we can use various different techniques that we have learned over the time to make this happen. And rather than cutting the gene off just as a straight cut here that I've shown with the red line, we're not going to do that because that would leave us with a problem. What we rather want to do, we want to leave a little bit to the very end, little extra. But we don't want that extra to be on both strands. So what we do something that we call as sticky ends. And let's have a look of that. So what we do, we use something known as restriction enzyme to perform this cut for us. So you can almost think of them as being molecular scissors that we use. So by adding these enzymes to a sample of DNA, it will find where we have coded, how we have prepared in a lab setting this enzyme to work. And based on the instructions that we have given to this enzyme, it will cut this original DNA strand off at 2 points. 2 points at the different base pairs. So that leaves us with this, what I described earlier on as a sticky end. We can use this sticky end for our benefit later on. And how we use it later on is that we use a method to carry this gene that we have cut off, and we take it where we want it to go. So it could be that other organism. This other organism could be simply another member of the same species, or as you might remember, it might even be to a different species. And to carry a gene from one organism to another, we often use something known as vectors. Vectors are often derived from bacteria. And what does this plasmid mean that I've shared here? What is a plasmid? So plasmid is in bacterial cells, what we find. Here's our bacterial cell. The DNA is often actually circular. I'm trying to get my pens out here, so bear with me. We find that the DNA is a circular, and we can break this circular DNA open, and then we can add this section of another gene that we were interested. So all of this section that I've shown here on the original diagram, we'll add it into that circular DNA of a bacteria, and we put it back into the bacterial cell. And then this bacterial cell could make many, many copies of itself. And in this way, we would have many copies of a vector that has this, this gene implanted into it. Well, then we put this, this vector into another cell. And now that cell then can do whatever we're aiming to produce out. Well, I kind of jumped ahead the gun with this explanation because one logical question that I assume that many of you would ask is that how does this piece of DNA that we just cut off attach to the vector? Well, vectors, similarly to what we saw with the DNA at the start, gets cut with sticky ends, and we use a material known as DNA ligase to join these pieces together. So there will be a similar sticky end also on the vector as there is on our gene of an interest. And we use the DNA ligase almost like as a glue, if you wish, to attach these two together. So I know that that's a lot of terms to kind of add to the discussion, but really the concept I think is best explained by thinking of a real world example. So what I wanted to do, I wanted to discuss with you about production of insulin. So as many of you know, we do sometimes have individuals who are not able to produce their own insulin. These would be individuals with diabetes type 1. And before we had these DNA techniques that I've just discussed with you available, what we had to do to gather insulin, which is an important medication that these individuals would take, we had to collect it from pigs. So we grew a huge number of pigs, and then they got slaughtered, and we extracted pig insulin from those corpses and then purified it and bottled it, and that was what was the drug that these individuals who can't produce their own insulin were then relying on. Well, you can imagine that this process is very wasteful because we are ending up going through a huge number of pigs. And there is also some risk of contamination at certain parts. There's more risk of things not going exactly how we planned. So what DNA technique has allowed us to do, it has allowed us to identify the gene within a human genome. So this cell that you see here on the left-hand side at the top would be a human cell. And within that DNA, within the nucleus of a human cell, there is a gene that codes for the insulin. So we can separate that gene that codes for an insulin from a human cell. And then we can take from a bacterial cell, and I'm just changing the pencils again. So here we have a bacterial cell at the top on the right-hand side, and we take this vector that we discussed about, and we cut it open, and we add into this the gene that we had that coded for the insulin. So the gene from the human genome that produces insulin. Now this vector can be put back into a bacterial cell, and we can take good care of these bacterial cells in a lab setting. We provide them everything that they might need: nutrients, warmth, and humidity. And all of a sudden, these bacterial cells are going to be growing in great numbers. So they're multiplying in a lab setting to large numbers. So this is how, from just one single cell where we added the gene for the insulin, now all of a sudden we have a huge amount of bacteria that all contain that human gene that coded for insulin. And if there's that human gene that coded for insulin, well, what these bacteria will do, they will actually implement the instructions from that gene, and they will produce insulin within the bacteria. Bacteria itself probably wouldn't need the insulin. It wouldn't be something that the bacteria really is needing. But if it has the information for it, and if we make conditions so that it wishes to produce that, now all of a sudden we have a lot of bacteria with a lot of insulin being produced in them. Then at that point, all we have to do is extract that insulin from the bacteria and collect it and bottle it and use it as a medicine for the individuals who might need insulin, the individuals that cannot produce their own insulin. So I hope that having discussed this process, it makes you think, if nothing else, about the fact that A, we're now saving a lot of pigs. B, we're able to do this in much more controlled manner. We know exactly what these bacteria are producing. We have less chance of contamination because we're doing this in a lab setting. So there's a lot of benefits, a lot of ethical aspects to consider, but also in terms of medical sense, a lot of things that make sense to produce insulin in this way. No one really that I have met has campaigned very strongly that we should save the lives of these bacteria. Bacteria are often single-celled organisms, so they really wouldn't be existing in a petri dish in a lab unless we grow them. And they don't have, to the best of our knowledge, any sort of sensation, nervous system, consciousness, nothing like that. So it's fairly generally well accepted method of producing drugs. And it's not just insulin that we produce in this way. There's a lot of other medications that are produced in this way. So basically what we do, we take a bacteria and turn it into a factory that produces whatever we want it to produce. And of course, there is a better fit because now this gene was from actually human genome rather than using another species insulin that's not produced based on the exact same instructions as what the human genome would contain. So I hope that that process makes sense now that we have had a chance to look at that through the example. And again, I know that these courses, I find these introductory level biology courses a little challenging because I'm telling you the big picture of the story. And you probably have a lot of little questions that you want to find out. Well, how does this happen? Tell me in more detail. And to really understand the little detail, we would need to spend significantly more time and go much more in depth into this process. So what I'm trying to do really today is to give you kind of an overview of the process. And that overview is aimed to give you more of an appreciation than a full understanding of every single little detail, but more of an appreciation why these DNA techniques that we have been discussing are so important and why they are so much more relevant than simple things that we saw examples of the garden peas. So, any questions about anything that we have discussed so far? Does everyone feel quite comfortable with that? I am assuming so. So what I want to do with you is to discuss a little bit about this thing that I mentioned earlier on the today's session, the method, the technique known as PCR. So PCR stands for polymerase chain reaction. And what we do with PCR, we take a specific section of DNA and we make a lot of copies of that. So the process of making many copies of a particular section of the DNA, that process is known as amplification. So PCR methods are used to amplify certain parts of the DNA sequence. And those parts are typically genes that code for something useful. And we do end up using few different tools in this process of PCR, but really the big thing to notice is that we are not copying all of the DNA. We're only copying the target, the gene that we're interested. And these machines that we use to do PCR are really not that complex. They are scientific machines, but the basic concept behind them is fairly simple. I mentioned to you earlier on that we can use heat to open up a double-stranded DNA. Then we can add certain enzymes that allows us to start making a lot of copies of that DNA strand that has been opened and copies only of the section that we're interested. And then we can keep doing this cycle after cycle, and with every cycle the amount of DNA sample or DNA in that sample doubles. So very quickly we're reaching great amounts of DNA in this initially small sample. So that's in a nutshell what PCR machine does. And most of the labs, most colleges, most universities, science labs do have at least one, if not more, PCR machines. And I think every single lab in science division at the CAC would have one. So that's something that I would have loved to show you if we had been in person on this class. And I think if we had been in person on this class, we would have definitely done some amplification of a DNA. So what I wanted to do next is to show you a video. And it's a little bit of a silly video, but there is a purpose behind that video. So this video is very 90s. It's a music video. But every single person that you see on that music video is actually, they are leading scientists working on the genetic field back at that time. That does include some of my former mentors as well. And what these scientists do, they sing a song about why PCR, polymerase chain reaction technique, is so useful and what we can do with it. And again, in my face-to-face classes, I do encourage my students to actually memorize this song to the extent that they get if it helps them to remember what PCR is, how it works, and what we can do with it. I am not asking you to memorize it word for word. I'm simply asking you to get the big message. However, in the past, and I'm not promising that it's going to be the case on this class, but in the past, I might have had an extra credit question in an exam where I have asked students to summarize what PCR is using this video to help them. So I'm just putting it out there. So I'm going to stop sharing for a second, and I'm going to share another screen with you. So what I'm hoping to do with you is that we take a little bit of time together to listen through this song about PCR. I am going to be honest, it is a little cringe-worthy in a sense that it is very 90s. But since I have asked about that in the past in the exam, it might be worthy of knowing. So if nothing else, this is a good chance for you to enjoy a little jam. Try to get from that, what do you think would be useful for knowing what PCR is and what it allows us to do.
There was a time when to amplify DNA, you had to grow tons and tons of tiny cells. And along came the guy named Dr. Carey Mullis, said you can amplify in vitro just as well. Just mix your tablet with a buffer and some primer. Nucleotides and polymerases too. Denaturing, annealing, and extending. Well, it's amazing what heating and cooling and heating will do. PCR. Detection, PCR, when you need to recombine, PCR, when you need to find out who they can use it, PCR, when you need to solve a crime, PCR, when you need to detect mutation, PCR, when you need to recombine, PCR, when you need to find out who the daddy is, PCR, when you need to solve a crime, PCR.
To all the scientists out there doing PCR, BioLab salutes you with the all-new 1000 series thermal cycling platform.
Well, that was quite something for a science class. So I hope you had a good jam and enjoyed that. Let's jump just back to our earlier PowerPoint. Let me just get it up. And you can definitely find, I think I've also shared that video in your learning material for this week. So if you really get into it, it certainly is one way to study for an exam. So what we saw, PCR was used to do quite a few useful purposes, or it was used for quite a few useful purposes in science. And we're going to look at few of these today. Before we do that, I think it's a good chance for us to revise a couple of questions. So again, if you do want to get those extra credit points for participation, this is your chance to share your answer in the chat. So what does PCR stand for? Is it A, polymerase chain reaction, B, phenotypic coupling recombination, C, phenotype correction reapplication, or D, P generation converse restriction? And that should have been a little, quite an easy question. So of course, we're talking about polymerase chain reactions when we're talking about PCR. And the second question that I have regarding the PCR is that, is it true that amplification refers to the step of the PCR process where many copies of a segment of DNA is made? Is this statement true? That's for A, or is it false? That's for B. So is amplification one step of PCR process, meaning the stage where we're making many copies of a particular segment of DNA? That segment could be, for example, a gene that codes for something. And if you answered true, you are absolutely correct. Amplification referred to making many copies. So well done for everyone who picked those up. I do appreciate your participation. Based on the PCR song, this technique allows us, and there's 4 options that I'm providing you. Remember, there might be more than one correct answer. Does PCR allow us to detect mutations? So notice if there's something wrong with the DNA. Does it allow us to do recombination? Recombining, we talked about that in terms of using a vector, for example. Does it help you to do parent testing? Who's the father of the child? And can it be used to solve a crime? So which ones of those do you think are true? What does PCR technique allow us to do? And if you answered all of them, you are doing really, really well. Remember, these are questions that might be on your this week's quiz. So one of the techniques where we use PCR is known as DNA fingerprinting. So just like your fingerprint is unique, so is your DNA. So within your DNA, we all have similar genes that code for similar things. But another thing that the DNA includes, it includes a lot of material that sometimes have been discussed or described as junk. DNA material that doesn't code for any particular trait or any particular protein. It's just in there. And people do differ in this part of the DNA as well. So the DNA is unique in that part as well. I think I heard a posting. I'm just checking if there's anything in the chat that I should have noticed. Oh, Amy got it correct. You're correct. Absolutely right. I'll make sure that you get a lot of extra credit for that. So we use this non-coding part of the DNA to identify that who does this DNA belong to. DNA will be similar amongst individuals who are closely related and less similar amongst individuals who are not closely related. If we find DNA from a crime scene, and then we collect a sample of your DNA, and if they are a match, we can tell that, well, somehow your DNA ended up to the crime scene. And one of the things that I've shared with you in the past is that I have spent some time in working on the forensic field, doing forensic work. And we never say that, oh, you're guilty because we found your DNA at the crime scene. Us individuals working in the forensic field, we're not in a position to make that statement. All we do, we can say that the DNA we found from the crime scene is a match to this individual's unique DNA, to this DNA that we will not find in any other individual on this planet, except if they have an identical twin. So what we do with the PCR technique is that we can take this small amount of DNA that we have recovered from crime scene. And I can almost guarantee you that if you've been to the crime scene, you have left some of your DNA in there. We all lose all the time a huge number of cells. We lose hairs, we drop things, there's droplets when we speak, as we've seen with the COVID pandemic. There's all sorts of parts of you that you leave behind even without realizing to every place where you're at. And that goes to the forensic field. We have a rule that every contact, every time you touch something, a little bit of you remains behind that object that you touched. So from a very, very small amount of DNA sample that we recover from a crime scene, we can actually then multiply the amount of the DNA that we found using PCR techniques. So this very technique that we discussed. And then when we have a lot of this DNA now, we use a system, we use a technique known as electrophoresis. And what this technique does, it separates segments, little parts of DNA, and it separates these parts of DNA depending on the size of this segment. And the sizes of this segment vary from one individual to another. So it's this non-coding parts of the DNA that really do not code any traits, but that are unique to you. And the size, the length of how many bases we have on that segment, that determines how far within a gel these segments travel. And this might sound a little bit of vague, but let's look at a set of pictures. And this is a picture of a DNA fingerprint that we have ran. We use very similar process that's fully automated these days, but we could do it fully manually in a lab. And that's what I normally do with my students. So how this DNA fingerprinting works is that we have a gel-like substance, so kind of like you would take a gelatin or something like that, and we make a sheet out of that. And within that sheet, we have small wells, so small depressions within there. And into these small wells, we put different samples. We put the sample that's from the crime scene, and we put the sample from various suspected individuals who we suspect that might be responsible for that crime. And then we run a sample by putting an electrical current through this gel from one end to another. And this electrical current makes these segments of DNA to travel from one end of the gel to another end. And as the current travels, this happens to all of these wells, samples from all of these wells at the same time. They all keep traveling. And if we were to have a sample that's from the individual that matches with the sample that we found from the crime scene, we would see same bands on these two samples. So what you end up doing when you're analyzing a DNA fingerprint, you're trying to find, is there something where there's a match with the crime scene sample and the suspected individuals for the crime. And there's going to be multiple bands. It depends a little bit from the country that how many bands are we looking at. But we're adding more bands, but it doesn't have to be huge numbers. If we find that they are identical, we can conclude that that individual with a very, very, very high percentage, higher of a chance, higher of a likelihood than it is to win in a lottery, this individual is the same individual than the individual from who we had recovered a sample at the crime scene. So that's one use of DNA fingerprinting, solving the crime as we saw. Another use would be if we have a child, and whenever you have a child, we're usually pretty certain who's the mother, just based on the pure biology. But we are not always sure who's the father. Well, if we do a sample from the child, and we do sample from the mother, and then we do the same thing that we saw from the suspected parents, there will be 50% of the genetic material of the child that's going to match to the father's bands. So related individuals will have similarity on the bands that we will see as a result of running a DNA fingerprint. So that's really the basic concept. I'm not going to be asking you to be able to explain this in a great detail. What's more important for me is that you see that all of this DNA stuff is somehow useful. I have a little bit of a better series of pictures. These are from a colleague of mine who took pictures as their students prepared a DNA gel, fingerprinting gel in a class. So what we're seeing here on the first picture is this gel. You really see that it's this jelly-like sheet that has been prepared. It's in a container that contains liquid, and you can also see that there's a tiny wells at one end of that gel. And this individual is using a micropipette to put a small amount of sample to each of these wells. So one well will include the crime scene sample, second well, sample from one individual that's suspect, second individual, third individual, and so on and so on. Or it could be that it's sample from a child and then potential from the mother and potential father candidates. Well, what we do then, we actually put this gel that's within this container, we put an electric current through it, and you remember this electric current caused the little pieces, little segments of DNA to travel from one end of the gel to another end. We have used in this picture a dye added to these samples. The sample would be normally just colorless, but it's sometimes easier to put the DNA sample into the wells if we add a little bit of coloring with it. Well, once we've run the electricity, what we'll end up seeing is that these bands travel different distances. And we usually use ultraviolet light to see these bands very clearly, and that is then how we analyze this gel that we produced. Again, I'm not expecting you to be able to explain this process step by step. I'm simply wanting to show you an application of these methods, why it might be useful to know about DNA. So based on everything that we've discussed, time for another quiz question. The method where we are comparing the unique DNA of individuals, what would it be known as? Would it be known as cloning, DNA fingerprinting, genetic modification, or genetic recombination? And feel free to answer in the chat. What is this method that we just talked about? And this method is, of course, going to be DNA fingerprinting. So I want to talk a little bit more, kind of referring to other uses of why we might need to do cloning and why knowing about DNA is useful. As we talk about that, I just want to remind ourselves of few basic concepts that are important for us to kind of keep on mind. So every cell in your body is going to contain a copy of all of the genes in you. Not all of them are active. The cells that are in your eye will not need to use those genes that code for a hair color, for example. That will not be used, but they regardless still include instructions for everything that's encoded into your genome, into your DNA. The only exceptions are sex cells, so the cell from the mother and father, and few cells that during the lifetime of these cells lose their nucleus. But nearly every cell in your body contains all of the genetic material for all the things that your body can be. So any cell, really, because it has all the instructions, could potentially grow into a completely new identical organism to the source of that, or to that initial cells. So the method that we use to utilize these concepts is known as cloning. And there's two types of cloning that we can do. So in reproductive cloning, we really want to produce an individual or cells of an individual that are going to be exactly like the original one. So for a long time, we thought that you could not clone adults. It turns out that it is possible. You saw with Dolly the sheep, we could clone an adult organism. Cloning as such is not a new thing. Cloning has been around for a long time. If you think of farming and choosing from the crops the characteristics that make, for example, a certain food product, a wheat, ideal, we have been doing that. We've been picking the garden peas that were the most delicious, or that were produced the largest peas, that had the peas that were looking most pleasant. So we have been choosing those characteristics when we have allowed plants to pollinate with each other. So really, plant cloning has used, and growing crops has used the concepts of cloning for a long time before we knew how to take a section of a DNA and move it from one organism to another. So cloning as such is not a new concept, a new idea. Now we just have a technology to make it more easily and make it more selectively. We did already talk about Dolly the sheep in last week's session, so I'm not going to spend time talking about that. But I'm going to bring you a question that's, again, from your quiz for this week. Is it true or false that nearly every cell in your body, with the exception of your sex cells, carry a copy of your entire genome? Is it true? And good job. I really appreciate everyone who answered. It is true. So nearly every cell in your body is going to contain a copy of your entire genetic makeup. And this is what we use in cloning. The second question that I have is it true or false that reproductive cloning is a completely new method in science? This is one of the tougher questions, but bear in mind what I emphasized earlier on. And good job for everyone who noticed that that's false. We have been using reproductive cloning when we have been choosing crops that are ideal, and we've been doing that for a long time now. So in terms of the therapeutic cloning, there are really two uses where we see therapeutic cloning being used mainly. First one is for research. So if we just want to learn about cells, and when that stem cell, stem cells are cells that can become any cell at all, when they start to specialize and become particular cells, let's say a cell that makes bone, or a cell that makes muscle, cell that makes nervous tissue. So we might want to research that. So that would be one reason to do cloning. Another time when we use cloning is when we use that to treat human illnesses. So if we knew that you have genetic error in your DNA, so you have a genetic disease, for example, if you were not able to produce insulin, the genes for your insulin would be, by mutations, by errors in the copy, they would not be working correctly. How cool would it be if we could take that gene for making insulin from another individual and put it into your body and have it to reach all cells in your body, or at least enough cells in your body so that your own body could now produce insulin? That is not quite the case yet, but I predict that we are working really hard, and one day in the near future, that will be something that can be done. Other options could be, for example, if an individual has a kidney and gets a kidney disease and that kidney then dies, doesn't work anymore, how cool would it be if we could take a cell from your body that has instructions for everything that you are, including how to make a kidney, and we could grow that kidney in a lab. And once it has grown in a lab, we could put it into you. That is very close of being realism again. So we're working hard to make these things possible. At the moment, we're having most success by using embryonic stem cells. So a lot of the research focuses on the stem cells, but as we saw with the dolly, it is possible to use adult cells, adult stem cells especially, to produce new things. So that's a little bit about why cloning is useful and why it might become useful. And I promise that this next slide is really the last slide that we'll have a look today. There's some terminology that I want to discuss with you. Again, if you have not seen it in the quiz today, it's not going to be in your chapter quiz either, but I think it's important that you recognize these terms as you move onwards in your study. So if someone uses these terms, it's not going to be completely new to you. So whenever we talk about field of genomics, we talk about studying genes, and we talk about studying all of the genes of an individual. There has been projects where we have mapped every single gene of a human individual. And this was a huge undertaking. This was a huge project to do. And no research group or no university could do it alone. So what we did instead, we divided human genome, so all the genes from a human body, into sections. We said that you have a look of that section, we'll have a look of that section. And then when everyone had analyzed their little section, we put all that information together, and a full human genome is now available free of charge for everyone online. So base by base, we have mapped every single base that makes a human genome. So why this might be useful, one of the things other than the examples that I have shared so far that we use this is if we're studying evolutionary history of organisms. So for example, by studying how similar our genetic makeup is, we can see that humans are very closely related to chimps. So we probably have with the chimps a common ancestor. So at some point in the evolutionary history, chimpanzees and humans were preceded by an organism, were perceived by a species that was the same. We can follow these relationships back further and further and further back and trace back to where life first started. We also are able to tell that there are species that are further apart from us than other species. For example, mice developed way earlier, separated in terms of their evolutionary line from us humans than chimps did. So we're able to tell how connected, how related in an evolutionary sense we are to other species. Couple of other fields, other terms that would be worthwhile to know. We can talk also not just study of the genes, we can study proteins. the field where we study the structure and function and how these proteins act, how these proteins produce certain traits. That field is known as proteomics. What I didn't share with you when I said that going through and producing a kind of a description of every single base that makes up a human and human genome, what I didn't tell you is that that amount of data, the number of those bases, is so vast that no one individual can ever read through each and every single one of those. So this is where now with the developments with the computer technology, we're actually able to use computers to calculate things, to find information that we're interested in, to do these comparisons between species, even to plan what a perfect sticky end for a particular gene would be. So that field has emerged, and that's known as bioinformatics. So it's combining the computer technology to do things that are useful for us in biology, in particular in the study of genes and proteins. So I hope that those examples, they are only some of the examples, but I hope if nothing else, those examples show that there is so much we can do with our understanding of genes. And there's probably going to be so much more that we are not even thinking of yet. So that really brings everything that I wanted to share with you today to an end. Like I said at the very beginning of this chapter, I do apologize, or this session, I do apologize that I'm a little behind with the messages. Please do not take that individually. Do not take that as a sign that I don't care or I'm not interested. I will get back to you as soon as possible, and you will not lose out because of the delay of mine. If it's been a time-sensitive thing, we will make sure that you will have a chance to get the time that you needed. Having said all this, that completes everything I had planned for us to do today, and we have actually covered everything that comes up in your chapter 14 and 15 quiz. And if you just bear with me for a second, if you could post your name to the chat, I will make sure that you get credit for having been present for the entire session. And that completes everything I have for today. I appreciate everyone who joined tonight or who watches this recording. I will stick around to answer any questions if anyone has. But if you don't have questions, I hope you have a very, very good rest of the night, and I hope you have a very good Thanksgiving. The quiz, like I said, only 10 questions. It should be easy, and you should be ready to go for your Thanksgiving and relax, and we'll come back to everything else after that and get you all set up with the exam. So that's after the Thanksgiving. But for now, thank you, have a good night, and I appreciate you joining. Thank you so much. Thank you. With that, if you do have any questions, I am here to answer those, but if not, I just hope you have a very good rest of the night. I will stop the recording at this time, and I will stick around to take attendance, but also if you have any questions, don't hesitate to just speak up.
Okay. Professor?
Yes, how can I help you?
I think I left you a message and you haven't got back to me, which is fine, because I know you're busy.
I'm so sorry about that. But tell me, what if I can resolve it now? We'll definitely try.
So it was like, I think on the last homework, there was like not a it was oh my gosh, I forgot. It was like,
was it about the mitosis and meiosis or something that the grading didn't
something
quite
like
work correctly?
where you Yeah, you have to like match it up or something, like
Yes,
across. It wasn't a multiple choice one. And
yes.
I know I got some, and it said one or two wrong. So, but it doesn't tell me which ones I got wrong.
I
So,
think that you probably didn't get anything wrong. I think that there is an error in one of those questions. And I'm going to be honest, I have read your question. I just haven't had a
oh,
chance to
okay.
investigate it.
Oh, okay. Okay.
So what
All
I
right.
will do, hopefully tonight, and if not tonight, I promise that's one of my very first things tomorrow morning. I will investigate that what happened there, and I will answer to you. More than likely, it's that there's an error in the
Mm.
grading system, and if that's the case, you will get full marks and everyone will get automatically full marks. If that is not the case, I will be sending you a message explaining exactly what was correct and what was wrong. But please know that it's very, very likely that there's just an error somewhere in the system.
Okay. And then also, I just wanted to let you know that I did turn in an assignment late.
Oh, that's fine.
Um,
If the
oh, okay.
machine accepted it, you're absolutely fine. I am normally never penalizing anyone for submitting late. I'm just very grateful that you submitted it. So don't
Okay.
worry.
Perfect.
As long as it went in, you're good. And I will actually, on the last 2 weeks of the semester, start chasing after if anyone has missed anything. I'll be like, hey, you missed this. Do you want to still submit it? But if you've submitted, you're good.
Perfect. Okay. That was it.
Perfect. Thank you so much. And like I said, I'm really, really sorry that I haven't answered to those messages. It's not that I'm ignoring you or I'm being
Oh,
rude.
I mean, I
It's
didn't.
just
Yeah, I didn't feel like that at all. Because
okay.
I know.
Good. Well,
Okay. All
good.
right.
Well, I
Thank
hope you
you.
have a very good Thanksgiving. If there's anything else I can help, I'm here. But if not, enjoy your break and enjoy knowing that you're pretty much done with this semester.
Okay. And then before exam for chapters 10 through 15, are we going to have like a study guide for it
Yes, we will do that next week
next
when
week,
you
right?
come back. Yeah. So next week
Okay,
on the
cool.
Monday, I will spend my Thanksgiving to write the study guide, and the next Monday we will go through it.
Perfect. All right. Thank you.
Thank you so much, and thank you for all your hard work on this class. I really appreciate it, and I have noticed how hard you work.
Aw, thanks. Are you
Okay,
gonna be at this school next semester? Do you
I
do you
will
do
be.
what do you do Bio 102? Or is that the next one?
I do. That's actually my real job to teach
Oh,
AP. So I offer that both in person and online. If you go to the class search online, you can search also with the name of the professor or you can look it up. I'm going to be honest that I don't remember exactly what days and what times. I think if it's an online class, it probably is asynchronous. So we don't have scheduled these classes like this. But even though I don't have it scheduled, I normally offer these classes still weekly.
got it. Okay. Okay,
Have
cool.
a look, and if you can't find something and if you need any help, just let me know. I really do think that you're doing amazing, and it shows that you're going to do really well in those classes as well. This is all the basic information you need to excel on those.
Okay. All right.
Have a good night.
You too. Take care. Bye-bye.
Bye.